Okay. Hello, everyone. Hope everyone's doing great. I know you guys are getting prepared for your final tomorrow. I want to do a quick review on neural codings because they are important and they will show up on your exam tomorrow. So let's get started. Let me share my board. So here's the whiteboard. Sure. Okay. Perfect. Uh, so we have five different neural codes that we introduced in the lecture. Um, so the f- um, the first one that we are going to cover is lateral inhibition. And what do we mean by lateral inhibition? It's really simple. It's not complicated. The neurons that get excited by light or by sound or wavelength or anything, uh, those neurons that are excited will inhibit the activity of their neighbor neurons. So if there's a neuron that is activated by light, it prevents its lateral neurons. That means the neurons which are next to it. Uh, it inhibits them to get excited, and this phenomenon makes us to see edges with different color contrast and so on and so forth. There are tons of examples and videos that are on YouTube with animation that you can compare and contrast and really understand uh, what the lateral inhibition is. But the main idea or the basic idea is that when a neuron is excited, it prevents its neighboring neurons to get excited. The next topic here that I wanted to talk about is the center um, of surround on center of surround cells. Um, cells, which this will ultimately leads to enhance of detecting lines. So let's go through it. How does it work? So when we have so our ganglion cells, um, when the light hits our cones, it goes to a bipolar cells, and then it hits the ganglion cells. Uh, our ganglion cells has let's do I'm gonna do I don't know white. Okay, so our ganglion cell has a on center, and we also have a I have to draw this, and we also have a off-center cells. So if we present a light to this area, to the center um, of the cell, this is the cell. So if we present a light to if I hold the flashlight to the center of this cell, what happened next is that the firing rate of that cell uh, it increases. If this was the baseline of no light presented, this would be the baseline of firing rate. Now, if I added a light to the center, then it increases the baseline firing rate for that cell. So we can see that it increases the action potential. So this is the increased action potential when I turn the light on the center. And then after I turn it off, then it goes back to the same firing rate, to the base firing rate. Now, what happens if this is the center, this is the off center. Now, what happens if I introduce a light to the whole thing? So if I increase my, you know, my light to cover the whole thing. So this what happened is that it actually diminishes the firing rate, believe it or not. um, so our firing rate, it goes down. Still there's it's more active, but it's almost close to the baseline. So maybe it's a little excited here, but it's really close to, it goes back to our baseline. So when for example, when we had no lights, right? Now what happens when let's see, I had different examples. So if there's no light, it's, we have a baseline firing rates. If we introduce a flash light to the center, we get excited, it increases the firing rate. If we introduce the light to the whole thing, off center and on center, it actually diminishes the firing rate, and it goes down to almost the baseline firing rate. And now what happens, the third and last one, what happens when I only put my flashlight on the surround and not the center? So what happens when I do just the surround and just I leave the center out of it? So this would, let's see. So this would actually suppress the firing rate when I turn on the flashlight. And then right after I turn it off, I see that, you know, it actually, I see a increased firing rate after I turn it off. So it actually, while the flashlight is on the off surround, I get no responses. But once I turn it off, I get the increased firing rate. And this is for the on-center and off-center cells. Let's go ahead and cover the next topic. Okay, oops. Let me do. Okay, now, what was the other one? We also had opponent process theory. So we have opponent process theory. This is for the color vision. Opponent process theory. This is the phenomenon. This is the theory for color vision. We know that for the trichromatic theory, it wasn't enough to explain everything that is happening, every color that we see. For example, the trichromatic theory didn't explain the image after image. And now the opponent process theory came into play, and they said that we have neurons, we have different neurons that works, the color vision works in an opposite way. So for example, I have a neuron, we have a neuron for a green color, and then the opposite of it is red. And the idea is that if I keep staring at the green, my neurons that respond to the green color, get tired, and then as a result, they fire less and less. And now, it's possible that, as if I look at a different picture, or different background, I get more feeling that I see more red, because simply the firing rate for the green was lowered, because I fatigued those neurons, and it's unaffected by the red neurons. And when I look at the like a white surface or something, I feel that I see for a second more red, because my green neurons are not firing as much. And it's the same thing as blue and yellow and all the colors that are out there. And this is the exact basic idea for the opponent process theory, that came to in order to complete the trichromatic theory that is out there. So these were the neural codings for vision. Now let's move to audition. And when we move to audition, we have a place, we have a place code for frequency or place coding. And this is covered in the lecture, but I'm just going to touch, a quick review, is that we know that this place coding theory explains that for our basilar membrane that is responsible for, for detecting the frequency that sends to the signals to the brain, that we ultimately hear stuff. The different frequency activate the different places on the basilar membrane. For example, if this is the basilar membrane, if this is the base, and this is the peak, oops. If this is the peak of it, different places on this basilar membrane encodes a different frequencies. Given that the base of the basilar membrane tends to receive more higher frequencies, and the peak of it receives more lower frequency, and this is the place coding. There's another phenomenon, or there's another the theory that came to the play, and this is the phase locking. This is actually in completion, or it actually emphasizes the Wally principle, but it's really simple. It says when the basilar membrane, if this is the basilar membrane, and this, when frequency hits it, it moves in motion, and it creates a, a wavelength. Then the neurons fire according to those, wavelength excitation and inhibitory. So we see here that the basilar membrane at this place was excited because of the frequency, and then neuron codes right here in regards to that phase. So this is really simple. So as basilar membrane moves upward, which is excited, and downward, the neuron codes the basically the same way. When it's excited, the neuron codes start firing, and then when the basilar membrane is its inhibition, then it's the coding for the neurons it becomes less. And yeah, this is all I wanted to cover. This was just a quick review that I wanted to go over and post it. As always, if you have any questions, any concern or anything, you can always email us, and we will respond back. We'll try to respond back as soon as we can. And yeah, that's it. Good luck on your exam, and hopefully I'll see you later.